All right, folks, so we're going to work on the mitosis-meiosis interactive activity. And I'm going to go through this with you and help you think through how to answer these questions. So the introduction here, this is for activity 1 called the cell cycle. The cell cycle is the repeating set of events that cells go through as they grow and divide. During the cell cycle, the cell grows, prepares for division, and divides to form 2 daughter cells. Each daughter cell then begins the cell cycle again. So at this station, you'll be identifying, labeling, and describing each part of the cell cycle. You'll first begin by answering the first question on your answer sheet, which is on the next page. The graphic organizer, which is also a diagram on that sheet, it represents the different phases of the cell cycle. You're going to follow the directions on the answer sheet to identify, color, and label each part of the cell cycle. And then you'll briefly describe the events of the cell cycle. So if we go down, we take a look. Here we have a nice diagram that shows different parts of the cell cycle. And your task is to complete the activity and questions on the student handout, which is the next page. I do have a link to the page in your textbook that will help you answer a lot of these questions as well. So let's click that. And you can see this is directly from your textbook. You can also have your textbook open to this. This is chapter 5, section 1. And so here's a diagram of the cell cycle. You can see the very outside area is called interphase, this whole part, which is part of gap 1, which is G1, S, or synthesis, and G2, or gap 2. And then you have the M phase, that's mitosis, and then cytokinesis. That's where you actually have the cell division. That's prophase, metaphase, anaphase, telophase, and cytokinesis. If you need to know what's going on in each phase, you have a very brief explanation here. But if you keep scrolling, it goes into much more detail on the next page in your textbook here. So you can actually see what happens more specifically in each of those different parts. So let's go back to our answer sheet here. The first question here says, what is the cell cycle? Well, I remember reading that just at the beginning here, where it says the cell cycle is the repeating set of events that cells go through as they grow and divide. So that's what I would suggest writing as your answer right in this area. So you can pause and do that right now. The graphic organizer below, here number 2, represents the different phases of the cell cycle. The phases of the cell cycle are listed below. You're going to use 10 different colors, if you can manage to figure those out, to color each part of the cell cycle. You'll color in the square to the left of each phase in the list below, and then use the same color to shade in the appropriate section of the diagram. Use your text tool to label the sections of the diagram. So here's what I'm going to have you do. There are a couple ways you can go after this. Remember how in that other picture in your textbook, the very outside layer was labeled? So you could always start with that. And you can type in, you have your text box here, so you could type in what its label is. And then you can use the drawing tool to figure out the colors that you want to use. So I'm going to do that first one for you. This first part is going to be called interphase. So we're going to type in interphase, okay, and then we'll pick a drawing tool. And what I suggest is if you go to the custom color wheel, there's actually 10 columns here. Pick a color from each column, and then you have your 10 different colors. So I'm going to go with the first one here. I'll stick with the bottom dark colors. I like that. So I think that's going to be right here. And I'm going to use that as my interphase color. So there's my color I'm going to put in a square. And then ever so carefully, I'm going to try and fill in the outside area. It's okay if it's not perfect. See how that it's going to be hard to do. And so there we have interphase done. And then you're going to go and pick another color for G1. And if you're like, I don't remember what G1 is, that's where you go back to your textbook. And you go back and you look at this picture. And you can see here, gap 1 or G1, okay, stands for growth, basically. G stands for growth. So cells are going to grow, carry out their normal functions, and replicate their organelles. So you go back and You can see if you look here, interphase, this is all of interphase, and it's divided into 3 sections, gap 1, synthesis, gap 2. If we go here, this is all of interphase, and it's going to be divided by gap 1, synthesis, and gap 2. And then that leaves the mitosis phase right here. Okay, so that's how you're going to divide it out. Now there's one extra phase here that we don't really see on this picture. But if you go back up here, you can see there's a G0, and it's kind of coming off of G1. And so if we go down here, you can see there's this arrow pointing off of G1. That's going to be your G0. Okay, so I'm going to help you out with that one right here. So G0 phase. Okay, and then you'll color the arrow, whatever particular color you want to color it. Okay, but I'll help you out with that one there, just because we didn't really talk about it in class. I didn't really, point it out at all, so that's where we're getting it from right here. So you're going to keep going with the different colors, coloring in this diagram to the best of your ability, and then you'll move on to some more questions here. So it says briefly list the events that occur in each of the following phases. Well, that's going to be easy enough if you go back to the textbook page and you scroll down. You can see all the different events that are occurring in that part of the cell cycle. So you'll just kind of go back and forth and write down the events that are occurring. Okay, so that's activity 1. Now activity 2 is looking at mitosis in plant and animal cells. So here's your directions. At this station, you'll be observing the stages of mitosis images as viewed under a microscope. Normally we would do this in class, but I've given you pictures to look at. You will observe how the phases of mitosis look in plant cells by observing the slide of an onion root tip. Onion roots grow very quickly, so there's a lot of mitosis going on in those cells. So this is actually a really neat area to look at on a plant. You will observe the stages of mitosis in animal cells by observing the slide of the whitefish embryo. So a whitefish is a very quickly dividing embryo, so we're going to look at a few of those cells too. Step 1 here, it says observe the image of the onion root tip. Spend a few minutes looking at all the different cells that you see. Then I want you to find a cell that is showing one of each of the 4 stages of mitosis. So you're going to find one that looks like it's in prophase, one for metaphase, anaphase, and telophase. You're going to label the stages using the drawing and text tool. Okay, and I'll show you how I would do that. And then you're going to observe, number 2 here, observe the images of the whitefish blastula. Spend a few minutes looking at all the different cells that you see and find a cell that's in each of the 4 stages, prophase, metaphase, anaphase, telophase, and label using your text and drawing tool. So here's your tasks. Identify and label the phases of mitosis. Describe the main events of each stage, and you'll do that on the answer sheet. And then you're going to compare and contrast a little bit between plant and animal cells. So let's go to the next page here. Here's some cells from the onion root tip. Now, the first thing I want you to do is go back and remember what's going on with the chromosomes in prophase, metaphase, anaphase, and telophase. Okay? You have to review what they're doing, because the dark parts in these cells are the chromosomes. So if you know where they're located or what they're doing in each of the phases, you should be able to identify them pretty easily. So if we start with prophase. Prophase is the first part of mitosis, and the chromosomes are going to be condensing. So you have still kind of a nucleus, but it's starting to break down. But then you're going to start to see clumping, and you're going to see actual chromosomes kind of clumped together. And it's going to look kind of like speckled almost. Normally in interphase you can't see anything, you can just see the nucleus and that's about it. But in prophase you're actually going to start to see the chromosomes. And then we have metaphase. Metaphase and middle, remember, start with M. So you're going to see the chromosomes lined up in the middle. This one's a pretty easy one to figure out. Anaphase, you're going to see them being pulled apart. The chromosomes are being pulled apart from each other. And then telophase, you're going to see kind of 2 separate nuclei almost, and you may see like a membrane or a cell wall coming between the 2. So it kind of depends on how far along in telophase the cell is. So let's go through looking at some of these. Let me see if I can find. Okay, I'm using this focus mouse button here. So here we have a cell where you can see there's like still just one nucleus, but you've got chromosomes that are becoming visible. If you go over to this one here, you can see it's very faint, and this is going to be a cell in interphase because the chromosomes have not condensed yet. So this is not starting cell division yet. This one is starting cell division. The chromosomes are condensing, so I would probably call this a prophase. And then we go down, and this one looks like they're moving apart from each other, and I would probably call that anaphase. And then if we go down, these next two cells, and this first one might be very early prophase, but I would still call both of these interphase. This cell here looks like I would probably call that prophase just because they look like the chromosomes are clumping and starting to condense. Here, the chromosomes are kind of lined up straight across the middle, so that's going to be metaphase. And then we have this cell here. Again, the chromosomes are condensing, so that's probably prophase. And then we have interphase because you can't really see chromosomes. And then I'm going to come over here. This one here looks like it might be in prophase because the chromosomes are condensing. And then here we can see that chromosomes are being pulled away from each other. That looks like it might be anaphase. This cell looks like it's just finished cytokinesis. So it's at the very end of telophase and cytokinesis. You can faintly see a new cell wall forming in between the two nuclei. These next few cells here look like they're all going to be in interphase. These two cells look like they're in interphase because they aren't really doing anything. You can't really see the chromosomes. Here, this is very early anaphase. The chromosomes are being pulled apart from each other. These next two, again, you can't see the chromosomes distinctly, so those are probably interphase. And here again, we have chromosomes being pulled apart, so those are going to be anaphase. And then I would say the rest of these are going to be interphase as well, because you really don't see individual chromosomes. This one at the bottom looks like interphase. This one here looks like it's in telophase. You've got the chromosomes, they're kind of uncondensing, so they're unwinding. You can't see individual ones anymore. It's not quite two separate cells. It's not really anaphase because you can't see the individual chromosomes like you can over here. So I would definitely call that one telophase. This one is, they're both going to be in interphase, but they've just finished dividing. You can see how they're smaller than some of these other cells. So that's how I know they've just finished dividing. This one looks like it's in anaphase because the chromosomes are moving away from each other. This one I would probably call prophase just because the chromosomes are condensing and they're all still kind of in the middle here. And then in the rest of these here I would say interphase. And these are interphase. This one here is probably prophase because they're condensing. This one looks like it's just finished cytokinesis, so you can kind of see the cell wall in between the two nuclei. And then I would say interphase. This one looks like telophase again because you can see the two nuclei, but you can't see the chromosomes anymore. They're unwinding. And if we go here, these are probably interphase. Here's a really good anaphase again because they're moving away from each other. These are all interphase. And then we have another metaphase. They're lined up in the middle. So you pick one for prophase, one for metaphase, one for anaphase, one for telophase. Okay. You can do this a number of ways. You can just take your drawing tool, make it a little thinner than that. I would do like a 4 maybe, and you can draw a line from the name of the phase to the cell, or you can like type. This one is anaphase. You can do something like that. So it's up to you how you want to label or identify these cells, but you're going to choose one for each of the phases. And then you're going to do something similar with the whitefish. There's fewer cells here, so we're going to look again, though, for the location of the chromosomes. And we also are not looking at brick-like cells. These are more round in shape. So you can see you've got cells over here. These three here, I would probably call them in like interphase just because you don't see individual chromosomes. They're not really doing anything right there. The one down here, you can see the chromosomes are moving away from each other, so that's probably anaphase. You've got these chromosomes lined beautifully in the middle. That's going to be a metaphase, as is this one here. These are nicely lined up in the middle. This one here, you can start to see the chromosomes condensing, so I would call this one prophase. And then here we've got these are moving away from each other. These are going to be a nice anaphase image. And then this one here is going to be telophase. You can just barely see the cell membrane is almost pinching into complete cells. And you've got the chromosomes, you don't see them individually anymore. They're unwinding and uncondensing. So that's a really good telophase image. So again, you're going to label one of each of the phases for prophase, metaphase, anaphase, and telophase. And now we have some questions to answer. And you're going to describe the events in each of the stages of mitosis. Kind of just went through that and in breaking down what each of the cells are doing. So you can go back and listen to what I was explaining and how I was identifying each of the images, and you can write in what's going on with the chromosomes in each of those stages. Number 2 here, it says you just described the stages of mitosis in the question above. Where does interphase and cytokinesis fit in relation to the 4 stages of mitosis? Well, if you don't really remember, you can go all the way back to activity 1. Because if you remember here, we have interphase that is here. And actually, if we go here, you can see then we have the mitosis phase. So remember, interphase is G1, S, and G2. Those are all 3 parts of interphase. And then mitosis is a dividing part. And if you're still confused by both these images that you've done, you can go back to the textbook picture. And so here you can see here's interphase, here's mitosis, here's cytokinesis. And maybe this will help you explain how interphase and cytokinesis are related to mitosis. Okay, I'm going to let you describe that relationship and which ones come in what order or which one follows the other. That's kind of what I'm looking for, is you to explain the order of them. And then number 3 here, it says the diagram below here, it's going to show the process of cytokinesis. This is how the cells actually become 2 separate cells in plant and animal. It happens a little bit differently between the 2 types of organisms. So you're going to explain what the differences are. So let's take a look at these 2 pictures. We have cytokinesis in an animal cell on the left and in a plant cell on the right. On the left, we have something called a cleavage furrow, where the cell membrane is just kind of indenting, and then eventually it separates into 2 cells. So it says a cleavage furrow is created using a ring of contractile protein, actin and myosin, which just kind of like a tie string kind of zipping through the middle there. The cleavage furrow reaches the center, and then the 2 cells are pinched apart into 2 separate cells. And they do this because all they have is a cell membrane. Over here, for the plant cell, you have these little bubbles. They're called vesicles. They move to the equator, which is the center of the cell, and they fuse to form this structure, this tubular structure across the equator. The tubular structures merge to form 2 plasma membranes, or cell membranes, of the 2 new daughter cells. So you have a daughter cell on the left and a daughter cell on the right. You can see where their nuclei are. And then you have pectins and other substances deposited between the 2 membranes, okay, in this area. And this area here is called the cell plate right now. And eventually all these things that are being deposited actually form 2 separate cell walls. And that's how you end up with 2 separate plant cells. They don't actually separate away from each other like animal cells do. They just form this cell wall in between. And it kind of goes from, one big cell, a cell plate is put down in the center, and then you end up with a cell wall for each of the 2 cells. So you're going to answer this question here, kind of explaining how plant and animal cells do cytokinesis differently by using my explanation and the pictures to write it down. And then moving on to activity 3, we're going to try and differentiate between mitosis and meiosis. So the directions, it says, are the stages of mitosis and meiosis the same or different? When looking at an illustration, can you decide if it is a stage of mitosis or of meiosis? So for this activity, you'll find cards, and they're on the next couple pages, showing each stage of mitosis and meiosis. And originally you were going to sort them on a tabletop, but here you're just kind of going to look at them and sort them in your mind. So you're going to spend a few minutes looking at the illustrations on the next few slides. Then you have to decide if the cards show a phase of mitosis or meiosis. You're going to look at the mitosis cards and then figure out what their correct order is. So after you figure out which one is showing mitosis and which group is showing meiosis, then you're going to try and figure out their correct order. Okay? Each of the cards has a specific code on them. You're going to record their order, okay, and I'll show you how to do that. And then you're going to do the same thing for the meiosis cards. All right, so those are your tasks. You're going to sort them into whether they're mitosis or meiosis. Then you're going to put mitosis images in order, and then the meiosis images in order, and then you're going to answer the questions. So here, is this mitosis or meiosis, and how do you know? So you're going to look at these 5 pictures, and you're going to figure out, is this showing us mitosis, or is it showing us meiosis, and how do you know? So first you're going to look at, okay, both processes we know start out with one cell. And then they duplicate, so in our prophase we're going to see chromosomes condensed. But remember the difference in prophase of mitosis and prophase 1 of meiosis. In prophase of meiosis, you're going to see the homologous chromosomes pairing up next to each other. In mitosis, they do not. And there's no crossing over in mitosis. And then in metaphase of mitosis, you're going to see individual chromosomes lining up single file. And in meiosis, they're going to line up in pairs. And then eventually they go through anaphase, where chromosomes move away from each other. And at the end of mitosis, remember, you're going to end up with 2 cells. And the end of meiosis, you're going to end up with 4 cells. So hopefully, based on what I just told you, you'll be able to figure out if this is showing you mitosis or meiosis. The next set of pictures, you have a few more. And again, based on the description I just went through and the process, you should be able to figure out if this is all showing you different phases of mitosis or all the different phases of meiosis. Then you're going to go back and figure out the order that these cells belong in. Which one is showing us prophase, which one is metaphase, which one is anaphase, which one is telophase, and cytokinesis. And then you're going to use these codes. In the corner here, you've got P3, C5, G6, and you're going to write them down. Actually, you're going to write them down on this next page on number 3 for mitosis, and then the one for number 4 is telling us about meiosis. Okay? After you do 3 and 4, you're going to tell me again, how could you tell the difference between the phases of mitosis and the phases of meiosis? How did you know the difference? And then you're probably going to want to pull up your notes from when we did the mitosis and comparing both of the processes, because there were some tables that really separated the two and compared and contrasted them well. So it's going to help you with these questions. So in what type of cell does mitosis occur? And then what type of cell does meiosis occur? Okay, so I've opened up my notes. They're the mitosis and meiosis lesson. You'll find these in the folders from before break. And so these are the notes that you're going to want to use to help you. So now this slide is a good one. It's going to help us figure out what type of cells divide through mitosis and what type of cells divide through meiosis. This is a good slide to use, so slide number 3. And if we go way towards the end, because I remember there being a comparison table here. Here we have a mitosis, what's going on in meiosis, what happens for both. So those are going to be helpful too. If a cell is undergoing mitosis and it contains 16 chromosomes prior to division, how many chromosomes will it have at the end of mitosis? Well, remember, if we go here, you're creating genetically identical cells in mitosis. So if you start out with 16 chromosomes, you're going to end up with 16 chromosomes, okay? And then referring to question 6, why would that be important? Well, you have to remember what is happening for mitosis. Why do cells need to go through mitosis? Well, in our bodies, we need mitosis for growth and repair. So why would you want all your skin cells, to make more copies of skin cells? Why do they need to have the same number of chromosomes? And then if you also think about mitosis can be used for asexual reproduction. So if it's used for asexual reproduction, you're making more organisms of that original organism, and they're exact copies. They're genetically identical. You're going to explain to me why would that be important to have happen in this process. Then question 10, it says if a cell is undergoing meiosis and it contains 22 chromosomes prior to division, before it divides, how many chromosomes will it have at the end of meiosis? Well, meiosis, remember, is creating sex cells. They're creating haploid cells, which means they're going to have half the amount of DNA, half the number of chromosomes. So hopefully that helps you. You can figure out 22 divided in half. You'll write down your number of chromosomes at the end of meiosis. And why is this important? Well, if we're making sex cells, that's the only way that we can maintain the correct chromosome number in the offspring. Because if you have 22 chromosomes in a sperm and 22 chromosomes in an egg, you're going to end up with 44 chromosomes in that new offspring. So really you want to have half the number in the sperm, so 11 in the sperm, 11 in the egg, and that way you can have the regular 22 chromosomes for whatever organism that is that we're talking about in this question. And then 12 is asking, what are the key differences between meiosis 1 and meiosis 2? So if we want to look at the differences between meiosis 1 and meiosis 2, we're going to need to go back a little bit, okay? So you can use this slide to help you take a look. You've got meiosis 1 here and meiosis 2. We can go back a little bit more. Here we have, nope, back a little bit more. Come on. Okay, so here we start with meiosis, and you're looking at the two sets of divisions here. Meiosis is going to reduce our chromosome number in half. The difference, though, one of the differences here, let's take a look at this diagram. Here's meiosis 1, and here's meiosis 2. This is a really good image to show what the differences are. And remember, in meiosis 1, crossing over occurs. It's not going to occur in meiosis 2. At the end of meiosis 1, you end up with 2 cells. At the end of meiosis 2, you end up with 4 cells. You do have half the genetic information and individual chromosomes at the end of meiosis 2. So I'll let you kind of pick out some of the major differences that you think you're seeing here between meiosis 1 and meiosis 2 for this last question. Now, if you've gone through and you've answered every single question, you should go up to this turn-in button and submit your answers to Schoology. And that takes you to the end of your assignment, and a job well done.